All right, good morning, folks. I'm Marty Downs. I'm the Deputy Director of the LTER Network Communications Office. Welcome to the 6th in our Spring Synthesis webinar series. Today we've got Lauren Hallett, Lauren Sheppard, and Jonathan Walter talking about integrating population and community synchrony across LTER sites. This is one of the 6 working groups that were funded in 2016 and 2017 by the LTER Network Communications Office. So they're a little less than halfway through a little more than halfway through their process. We'll get an update and the beginning of very interesting results out of the group. Lauren Hallett is an assistant professor at the University of Oregon, working on plant community ecology and restoration ecology. She has her PhD in ecosystem science from UC Berkeley and did postdocs at Berkeley and at UC Boulder. Lauren Sheppard's a postdoctoral researcher with the Kansas Biological Survey. PhD is in physics from Lancaster University, where he worked on using wavelet analysis to understand variation in intracranial pressure. He's now applying those tools to fluctuations in ecological communities. Jonathan Walter's a postdoctoral research associate at the University of Virginia with the NatureNet project. His research focuses on understanding ecological patterns in space and time, especially spatial synchrony and the population dynamics of outbreaks and invasions. We'll get started with these guys in just a minute, but I wanted to we've added one additional webinar to the series. We were going to wrap up and take a summer hiatus after this webinar, but we've added one for on the topic of a disturbance framework, a sort of reassessment of a disturbance framework. The idea for this came out of the 2017 LTER Science Council meeting, and Evelyn Geyser, John Kamenowski, and a group of other LTER ecologists are putting together a workshop for the LTER All Scientists meeting, using long-term data to validate this updated framework that came out of the Grimm et al. Paper. And they really want some help from the community in thinking through how that applies to LTER, the data coming out of LTER sites. So that webinar will be sort of at the same time as usual, July 12th at 11 o'clock Pacific Daylight Time. It's worth visiting the website and taking a look at some of the introductory materials in preparation for that. In terms of logistics, participants are all muted when they join. If you have questions, you can either ask them in the chat window, I'll be monitoring that, or there's a Q&A box at the bottom of the Zoom window. There's a little box that says Q&A towards the left-hand side. If you just click on that, you can type your question in, and I'll be monitoring those throughout the presentation and get them to the presenters. And with that, Lauren and Lawrence and Jonathan, thanks a lot.
All right. Thank you, Marty, and thanks everyone for being here. We're excited to talk about some of the ideas we've been generating and some of our preliminary results. All right, so the goal of our working group is to integrate population and community synchrony across the LTER sites. Again, Lawrence, John, and I are going to be presenting, but this is definitely a team effort of around 16 of us. And so in terms of what we mean by population versus community synchrony, so population synchrony is also known as spatial synchrony, and this often occurs that you have populations of the same species that are spatially distributed, but their populations fluctuate in tandem across space. And so in this kind of little dummy image, we see that we have 4 different patches, but the abundances of those populations are all fluctuating in synchrony, causing the abundance of the species across the landscape to have very variable abundances over time. And this is thought about a lot One, because of pest outbreaks. So you think about the proverbial swarm of locusts, or if you think about gypsy moths or pine beetles, often their populations are fluctuating in synchrony over space. And spatial or population synchrony is also of conservation concern. So if you have populations that are very synchronous across the landscape, when all of those populations are in decline, it can put them at greater risk of extinction. And so those are kind of the two reasons why this phenomenon has been pretty heavily explored. And there's been a lot of work in spatial synchrony about what are those mechanisms that are driving those patterns of populations fluctuating in tandem across the landscape. In terms of community synchrony, in contrast to spatial synchrony, community synchrony is often thought about within one place. So within a patch or within a community, the concept here is of synchrony among species. And so in any given place, if there's multiple species in that community, if those species all fluctuate in tandem, that would be what we would term community synchrony. And in this case, the aggregate of all those species fluctuating in tandem is that the community overall can be highly variable, and any ecosystem property that those species contribute to, be it productivity or cover or nutrient cycling, might also be very variable. And so community synchrony is often studied in the context of ecosystem stability. So thinking about things like production systems or productivity over time. If you have high community synchrony, you would expect more variable production, versus if there's asynchrony or anti-synchrony and species trade-off, it might be more stable. And so the goal of our working group is to integrate these approaches to population and community synchrony. So they're related phenomenon, but the tools that have been used to explore these different forms of synchrony are very different, even though there's a lot of kind of crossover of ideas and potential synergies between them. And so in order to kind of think about how population synchrony can learn from community synchrony and community synchrony from population synchrony, we assembled a working group that is roughly broken into 3 groups of people. So we have a set of quantitative ecologists, and many of these have been focusing on spatial synchrony, some on community synchrony. We have a set of terrestrial ecologists, a lot of them experimental, that are exploring these questions, but empirically in experimental communities. And then finally, we have a set of aquatic ecologists. And so a lot of these kind of ideas of synchrony, a lot have come from aquatic systems because the time series lengths have been long enough. So we're just at the stage where we have a lot of terrestrial data sets, like the grassland sites, for example, were started in the 80s. So they're just long enough to use some of those tools that population synchrony people have used in aquatic systems for a long time. So we're bringing together kind of aquatic and terrestrial data with this team. And when we started the working group, because we had people from such different perspectives of what synchrony meant to them, why they thought it was important, we did this exercise where everyone presented basically, here's what synchrony means to me, this is how I measure it. And just to give you a flavor for our different backgrounds, Lawrence and I are going to do that for you now. So I'm going to hand it over to Lawrence to say, from the perspective of population synchrony, what does he mean by synchrony, and how would he measure it? Lawrence.
Hi. So my approach to synchrony is based on the use of wavelets, and we use this in both population synchrony and community synchrony latterly. And this approach is designed to break down fluctuations in data by timescale. So we can examine the long timescale fluctuations separately from the short timescale fluctuations, and learn more about what's actually driving the changes in the community and in the populations. The way we do that is using these wavelets. So this image that you're seeing is an image of a Morley mother wavelet. And if you just look at the blue line there, you can see that there's a kind of sinusoidal fluctuation there, which is localized. So it starts with no fluctuation, and then there's a localized fluctuation, and then it goes back to 0 again. And what that's representing is this Morley mother wavelet, which is a complex fluctuation confined inside an envelope. And we can stretch or compress this wavelet so that it has a different time scale of fluctuation. This one has fluctuation which goes from low to high and back to low again with a scale of 2 units on this axis, but we can stretch it to have any scale we want. And we use those kind of test functions to interrogate our data. So on the next slide, which hopefully Lauren can bring up, you'll see a very classic example of fluctuating data. This is a lynx population in Canada, taken from the famous Hudson Bay data set about population fluctuations in Canadian lynx. And you can see throughout the 19th century, you've got this very clear periodic fluctuation. And so what we do is we perform a wavelet transform on this data, where we use wavelets of different timescales, and we convolve the wavelets with the time series, and that spits out a series of values that tells you how well the time series matches up with a wavelet of that time scale at that particular time. So if you look at that matrix of values on the right, you can see that time scale is on the x-axis and the date is on the y-axis. And there's that big yellow feature running through the data a time scale of about 10 years. So the wavelet transform magnitude there is telling us that there's a lot of fluctuation at the 10-year time scale, and that it continues throughout the whole of the 19th century, throughout that whole data set. Whereas the paler blue and the dark blue values are saying the fluctuations at other timescales are relatively depressed. And you can see that also that matrix has been kind of scalloped away at the top edge and the bottom edge. So there are missing values at the edges where the values are considered unreliable. So that's how you use a wavelet transform to extract information about the timescales of fluctuation. And it's really obvious in the lynx population because that's a classic sort of case. But we can apply that approach to any data we like and extract both this magnitude of oscillation that I've showed you that tells you what timescales the fluctuations are at and how they vary over time. And we can also extract what's called phase data. And going to the next slide, the phase information that you get about a fluctuation is telling you what stage of oscillation has been reached at a particular point in time. And when I talk about synchrony, what I mean is we've got data from 2 different sources, whether it's 2 different populations, so we're talking interested in spatial synchrony, or whether it's actual populations of 2 different organisms in the same location, so we're talking about community synchrony. And if those fluctuations maintain a particular phase difference, looking at the phases extracted from the wavelet transform, then we can say that there's some tendency to preserve a particular relationship between the oscillations of those 2 time series. And the classic case I'm showing you here is the fluctuations in lynx population actually follow the fluctuations in their food source, which is the population of snowshoe hares. So the snowshoe hare population peaks and then crashes, and the lynx population peaks and crashes just slightly behind that. So we would translate that as there's a phase relationship between the fluctuations on that time scale, and there's actually a phase shift between the hare and the lynx, such that the lynx fluctuations follow the hare fluctuations. So from now on, I'll be talking about phase relationships between data as extracted from the wavelet transform, and that's how we examine the synchrony on different timescales.
Great, thanks, Lawrence. So that would be Lawrence's answer to what synchrony means to him, why he cares about it. I'm coming from kind of the opposite perspective of thinking about community synchrony. And so just gonna give you kind of a taste of how I answer this question. And then what we're gonna do is walk through a couple avenues we've been exploring for how integrating these different perspectives is giving us new insights into communities and synchrony. So from my perspective, I often think about community synchrony because of its link to stability. So that idea that if you have species in the same community that are fluctuating in tandem, that can mean that ecosystem properties are highly variable. So in this dummy, we have 3 species all fluctuating together, causing a very variable aggregate community. But if you have some species that are asynchronous or others that are even anti-synchronous, so that would be this blue versus the tan species, those trade-offs can mean that abundances of the total community are are more stable over time. So thinking about this a lot from production systems. I work in rangelands. We think about it during cover, during drought. Are there some species that can essentially compensate when others decline? So often we would term this compensatory dynamics when there's trade-offs among species that are stabilizing. And just kind of give you a visual of a site that I work at where this phenomenon seems very strong. So this is a serpentine grassland in California. In 2001, it was very much dominated by forbs. By 2005, it was definitely a grass year. And so those forbs had really kind of been replaced by grasses. And then back by 2009, it was a forb year again. And those time series look something like this. So our dominant species, this plantago, is fluctuating at fairly long time series time scales. And then you have some trade-offs. So for example, if you look here and coming out of 2000 and before 2010, you see the plantago is high and then crashes and then is high again, whereas the bromus, which is the dominant grass at the site, is really kind of compensating for that decline. So think about those trade-offs between species, how we characterize it, and whether it's stabilizing. And so in my world, one of the most common metrics of synchrony is the variance ratio. And basically what this does is it looks at the variance of the overall community versus the variance of individual populations. So that variance of the community is basically looking at the covariance between every pairwise set of species in the community and relates that to how variable those species are in and of themselves. And so in a world where there's a lot of negative covariance among species, you're going to get a variance ratio that's less than 1 that would indicate compensation. And when there's a lot of positive covariance among species, the variance ratio is going to be greater than 1, indicating synchrony. And the variance ratio is one metric. There is another that's been developed by Leroux and Claire de Mazencourt that looks at covariance in a slightly different way. Another by Nick Gross that looks at correlation. But basically, the commonality of all of these metrics is that you get one value for the whole time series that looks at either covariance or correlation. And in this world, so what synchrony meant to me coming into the working group was that synchrony meant positive covariance between species and compensation meant negative covariance. And kind of one of the classic papers that explored this was actually from a previous NC's working group that basically looked at a bunch of different sites, the proportions of the subplots in those sites that had positive versus negative covariance. And so what they found was that most of the time, there was less than half of the sites had plots within a site had negative covariance, and suggested that maybe synchrony was more common than compensation across these sites. And these metrics have been used a lot. I've used them and seen differences across grasslands for what types of grasslands exhibited negative covariance versus positive covariance. It's been explored a lot. And kind of in my answer to that question is what is synchrony? It would be basically these metrics. But that segues into the kind of the first lesson that I would say the population synchrony brings to community synchrony. And that's to really start exploring timescale specificity of community synchrony. So what we're going to do for the rest of this webinar is I'm going to give one example of how we've brought timescale specificity to community synchrony. Lawrence is going to bring another example of methods that highlight a different aspect of timescale specificity for community synchrony. And then John is going to talk about what lessons community synchrony has brought for spatial synchrony and thinking about the spatial synchrony of community properties. And so if at any point, I think what we'll do is at the end of each lesson, we'll solicit questions, and then we'll do more questions at the end. So if you do have questions, do put them in that Q&A box or ping Marty and she'll let us know. So for thinking about timescale specificity of community synchrony, one of the first approaches that our group used was basically to take this variance ratio, but do a spectral decomposition to make it timescale specific. And so if we revisit that classic metric of the variance ratio, it is not timescale specific. You get one value for the entire time series. But what we did was we essentially replaced all of the variances with a power spectra and all of the covariances with the co-spectra that allowed us to partition dynamics of the variance ratio at long versus short frequencies. And I want to walk you through some reasons for why we did this from some simulations that was led by Lauren Shoemaker, and then go into some empirical results that we found from this approach. But the first reason we thought, it's really important to bring community synchrony up by different timescales is that different drivers can operate at different timescales. So we're going to be revisiting the Jasper Ridge example a bit throughout this talk for continuity, but Jasper Ridge is interesting that there's gopher disturbance that often happens on pretty short timescales. So in this particular example, we thought, you might have two species that are responding in the same way to the short-term timescale disturbance. So in that bottom panel, it's a black versus a gray species. And then their aggregate biomass is pretty variable over these short timescales in relation to this short timescale driver. But you also might have long timescale drivers. So a lot of the work coming out of spatial synchrony suggests that long-term weather patterns, such as the Pacific Decadal Oscillation or the North Atlantic Oscillation, can shape a lot of spatial synchrony. And you can imagine a community where two different species have different responses to these long-term drivers that might end up being fairly compensatory over long timescales. And so if you aggregate these, and so you think, both of these drivers are operating simultaneously, you get sort of the composite dynamics you see in this bottom right-hand panel, where there's some asynchrony or compensation occurring over long timescales, but synchrony over short. And so what we did was to kind of come up with a model that generated these different dynamics, and then applied our timescale specific variance ratio to them. And what that allows us to do is really partition the fact that you have positive covariance occurring over short timescales versus negative over long. So in this bottom panel, again, a variance ratio greater than 1 indicates synchrony, less than 1 indicates compensation. And in this kind of model that we've developed, what we see is that it allows us to really partition that there's synchrony over short timescales and compensation over long in a way that you just miss if you just looked at the classic variance ratio. And I think this is important for a couple of reasons. One is that these dynamics might be very ecologically real. And so it's important to kind of partition this to understand those dynamics and then think about drivers. Then second, a lot of the experiments we do to investigate synchrony versus compensation are often on the shorter timescale length of things. And so it's possible that if we're applying these classic metrics to too short of timescales, we're going to miss dynamics that might be occurring over longer timescales. So that was kind of rational one for why we thought it was important to decompose measures of community synchrony by timescale. And the second reason that we thought it was important was that different species can operate on different timescales. And so you can imagine that if you have a species that has a very high reproductive rate, so insects or maybe annual plants, they might have a growth curve that kind of overshoots the environment. So in this upper panel, that would be the pink species, versus you have some species that may be able to really track their environment, so that would be the black. And then other species that maybe have a bit of a lagged response to the environment. So it might be perennial species that just have a slower growth rate and respond a little more slowly to the environment. So if you imagine that you have different species with these different growth functions, that can affect the degree of synchrony or compensation that occurs even when they have the same response to the environment. So in these next kind of examples that we've developed, both species are going to share the same response to both of these drivers. So in our first kind of simulation, we have two species that both can track the environment at pace. And so we have this solid black species, the dotted black species. They both have that black growth curve such that they're tracking the environment kind of immediately. And what that means is in this world, you have very high synchrony among the species for both short and long time scales. Whereas in the second simulation, you have one species that tracks the environment, but another that has a lagged response. And so in this community, what you see is that lagged response means that you you end up having very high negative covariance over short timescales. So even though they're responding to the same environmental drivers, because of that lagged response, it ends up being kind of a compensatory dynamic. And then finally, if you have a community that has one species that really tracks the environment and another that can overshoot, that has a very high growth curve, what that means is that on the short time scales, they're both very synchronous, right? They're both responding to these short time scale drivers. But over longer time scales, the internal dynamics of that species with the very high growth rate dominates as opposed to the environmental response. And so you see more compensation than you would expect if they both track the environment at the same time. So these kind of simulations that were led by Lauren Shoemaker really highlight for us why it's kind of important to try these approaches, and also gave us kind of a platform to have expectations for what we see within real time series. So to that end, we applied our spectrally decomposed variance ratio across LTER grasslands. And we did this in part because a previous synthesis that I had led suggested that in areas with high climate variability, there was a lot of negative covariance. And in areas with more stable rainfall, there was often more synchrony. And so we thought, okay, can we decompose these patterns into their relative frequencies? And so this is some work that was led by Lei Zhao and also Xiaopeng Wang. And just to walk you through basically what these graphs that I'm going to show you are, is that the black points will be the variance ratio overall of the whole time series for a site. And then we're going to have the blue is going to be the variance ratio for short time scales, and the red for long. And so this is from one example site. This is one plot at Jasper Ridge that really exhibited these differences in variance ratio over short versus long time scales, such that there was a lot of synchrony over short and compensation over long. And so this is kind of the summary results that we found across 6 of these different grassland sites. And so on the blue, on the left-hand side, we have essentially the variance ratio over short timescales versus the red over long. And what you can see is for some of the sites, there was no difference. So the 4 left-hand panels. But for Jasper Ridge and also for the Kellogg Biological Station, you had this very pronounced pattern that there was definitely more asynchrony or compensatory dynamics occurring over these long time scales than over short. And so what we're doing now is kind of trying to look for the mechanisms that might be underlying this, and also thinking about the implications for stability. So that's kind of lesson 1, or approach 1, is a spectral decomposition of the variance ratio. Are there questions on that? I don't see any in our chat box. All right, in that case, I'm going to hand it over to Lawrence. That's approaching the same idea of timescale specificity and community synchrony, but applying a wavelet coherence approach. Lawrence?
Hi. So the idea of wavelet coherence, as I alluded to before, is the idea that the phases that we get out of this wavelet transform approach might bear specific relationships to one another, and we can show statistically that 2 or more time series are actually in phase or out of phase, or have some specific phase relationship when it comes to their fluctuations at a particular time scale. And if you've got more than 2 time series, if you've got more than just links and hair, if you've got, say, a bunch of spatial data, we developed an approach called a wavelet phasor mean field. So this is a way of taking wavelet transforms, taking a large number of wavelet transforms, and in this example I'm going to show you is an example we published in Nature Climate Change, where we took time series representing phenological variability in aphids in the UK, which was essentially the date of first flight of the aphids in each year, which was measured at different points in the UK. And we were examining their spatial synchrony on different timescales by making all these wavelet transforms. You can see all the wavelet transforms on the left, extracting the phase information at every time and timescale from those wavelet transforms, and combining it to make a wavelet phasor mean field, which is the thing that you see on the right. And that wavelet phasor mean field is high, or yellow, whenever all the transforms agree about the phase of fluctuation at that particular timescale, at that particular time. And it's low, or blue, when they all disagree. So you can see, reading this phase or mean field plot in the same way that you would read a transform like the Lynx transform I showed you before. Pick a time scale like 0.1 is one cycle every 10 years. Look at that time scale and trace it through the years of measurement. And you can see that 0.1 cycles per year and around 1988, there's a kind of big yellow maximum highlighted with the letter B. That means that all of the time series drawn from the different locations agreed about the phase of the 10-year fluctuations in 1988. Whereas you can see up in the top right, that point labeled A in the sort of blue area, that's a much shorter timescale fluctuation. That's 2 or 3 year fluctuation. And you can see that around sort of 2000, 2005, all the different transforms were in total disagreement about what the phases of fluctuations were at that timescale. So those short time fluctuations were really doing different things in the different locations. So in the next slide, you can hopefully see that when we took an average of that wavelet mean field of phenological synchrony for a bunch of different species, and we compared it to the wavelet phase or mean field that we got from looking at fluctuations in winter temperatures at those different locations in the UK, we see that the patterns match up very closely, and we can actually understand the variability in the spatial synchrony in terms of the variability in this driver variable, the winter temperature. So essentially, in the cold winters, the aphids fly later. And as a result of that, when the winter temperatures are synchronous across the UK, the aphid emergence times become synchronous. And that leads to a variability which you can hopefully see in the left-hand plot. In that bottom left quadrant of the plot, you can see there's a lot of synchrony at those long time scales in those early years, but that fades out as a result of the reduction in winter temperature synchrony. And then in the latter period of measurement, there's relatively much more synchrony at short time scales. So there was a changeover in the time scale of synchrony for the aphids, and that manifests in the individual time series, and it manifests in the kind of spatial aggregate behavior of the population. So that approach to spatial synchrony that we applied to the aphids, we're now applying to community data for this project. So in the next slide, you can hopefully see the general conceptual approach that we took to community synchrony was thinking in terms of a portfolio effect. When you have multiple species in a community and they're all fluctuating, you anticipate that if there's no phase relationships between those fluctuations, as you add more species and as the community becomes more even, the total productivity or total abundance of all the organisms in the ecosystem should be more stable as a result of adding more species, because the fluctuations basically tend to cancel out. And then down on the bottom left, you can see an extreme example of that is when you have fluctuations in antiphase with one another. So if you imagine time series drawn from two species that are maybe competing with one another or for some other reason tend to fluctuate in antiphase, that means the peaks in one line up with the troughs in the fluctuations of the other. That's a compensation effect, and that means there's excess stability in the community. The community as a whole becomes more stable than it would be just if the phases were completely unrelated to one another. And the opposite case is on the right. If you have a number of species all fluctuating in phase, like they're all possibly subject to the same environmental driver that causes them all to peak and crash at the same time, that's community synchrony, and that leads to instability, and the overall productivity of that ecosystem would tend to crash and peak in a very sort of obvious way, driven by that synchrony. So just as a test case, in the next slide, I'll show you that approach, that wavelet mean field approach applied to Jasper Ridge data. So 20 different species drawn from the Jasper Ridge grasslands, and combining their wavelet transforms in this wavelet phase or mean field, we see that there are some very bright, so yellow, white here is high synchrony, and red is low synchrony or compensation. And these values are compared to a null distribution of the distribution of values we would expect if there were no phase relationships between all these different species, and they were just kind of fluctuating independently of one another. That would be just a pure portfolio effect. you can see that those bright whitish yellow features, there's one at around 1995 at a timescale of around 2 or 3 years. There's one at around 2004 at a timescale of around 4 or 5 years. Those are fluctuations that have occurred in the whole community where all the different species agree about the phase of fluctuation. They've all gone high-low together. And those are big, unstable fluctuations in the community on short timescales. Whereas you can hopefully see up at the top of that plot, in around, let's say, 1996, 97, a timescale of around 11 years, there are some red features which are also marked out as being significant relative to that null hypothesis of no relationship. But those are significantly low wavelet phasor mean field values, which means that there's complete disagreement about the phases. There's more disagreement that will be affected by chance, which is compensatory dynamics. And that indicates that on those long 10 or 11 year timescales, there's actually excess stability driven by antiphase relationships between individual organism populations in this particular site. And before I hand over, I just want to point out that although we focused on the grasslands and this specific Jasper Ridge example, we're also applying this approach to a bunch of other community data sets, including a bunch of other grasslands, but also lake data sets and ocean data sets, including all kinds of things like plankton and coral and fish. So this is a generally applicable approach to understanding how the portfolio effect is actually manifesting in these communities and how compensation and synchrony are kind of competing with one another to produce either community stability or instability. And I'll hand over to John, I think.
Awesome, thanks. Before we switch to John, Lawrence, we had a question,
which
was how do we handle blocks of missing data?
How do we handle what, sorry?
Blocks of missing data.
Blocks of missing data, okay. So that is a general problem. Essentially, in order to take this timescale specific approach, which is all based on Fourier transforms, both the timescale specific variance ratio that you saw before and this wavelet transform approach are really fundamentally based on a Fourier spectrum. And the way that a Fourier spectrum works is it always works with regularly sampled data. So if you have missing data, it's really unavoidable that in some sense you end up having to interpolate that missing data. And there are wavelet approaches based on essentially not considering the missing data. And that's more or less equivalent to assuming that the fluctuations in the data that are missing, or assuming that there are essentially no fluctuations in the data that is missing. Sometimes you might want to do a smart interpolation of the data where, for example, if you're working with seasonal data and you know that there's a strong seasonal fluctuation in the data, it will make sense to do an interpolation where you replace missing values with what a typical value would be for that time of year, something like that. Or if you really have no idea, you might just interpolate through using a spline or just using an intermediate value between the values that you do have. But essentially, you have to fill in that missing data. And as a result of that, you sort of inevitably lose some statistical power. And then it's just a question of using statistical testing techniques to establish whether you actually have a significant result, whether you actually have a statistically significant phase association in the data that you do have after the fact of having to interpolate any data that's missing. Does that help?
I'm not sure they can respond, but thanks for that. Are there any other questions, Marty? All right.
Nothing yet, but give people a minute to jump in if they have them. Okay.
All right. So the other kind of general piece that we've been looking at is lessons from community ecology for the population and spatial synchrony. And John is going to describe some of the patterns we're seeing when we look at the spatial synchrony of community properties. John?
Yeah, thanks, Lauren. So yeah, this is a little bit different than the approaches that you've seen previously, but another way that we're looking to kind of merge these different perspectives on synchrony. I'm sure you recall from earlier in the webinar, when we talk about spatial synchrony, we're talking about fluctuations that are correlated through time across multiple locations. And traditionally, this has been studied at the population level. So the key ingredients for any typical spatial synchrony study are time series of abundances or population growth rates for several locations. But from a mathematical perspective, those time series don't need to be about abundances or growth rates, and they don't really need to be about populations at all for our ways of quantifying synchrony to work, whether that's a correlation coefficient or a statistic based on wavelet analysis. But of course, how we interpret what that synchrony means is going to change depending on what that variable is that makes up our time series. So what we wanted to do was to ask what happens if we replace those time series of population level variables with attributes of communities. So we can measure for communities time series of properties like richness and abundance and see how those change through time and whether those particular properties fluctuate synchronously across different iterations of a community type in different locations. And this seems to be something that people haven't really done before. It seems simple, and on the one hand, there's nothing completely new under the sun. But among a room full of 16 specialists in population and community ecology, no one had seen this done before. So it seemed like a cool way to try and again bridge these different perspectives, and hopefully develop some new insights into community synchrony. So one question you might have is why we would even think that community properties would exhibit spatial synchrony? Well, for one, communities are collections of populations. And so what happens to the populations that make them up must in some way have an impact on the community. And at the same time, spatial synchrony at the population level is nearly ubiquitous. So we could just conjecture based on that at least in some cases and to some degree, that synchrony would percolate up from those populations to impact community dynamics. And we also know that some of the major mechanisms of population spatial synchrony are also important for communities. As Lauren and Lawrence have both mentioned, compensatory dynamics can result from different members of the community responding differently to environmental perturbations. These same types of perturbations that can produce synchrony in populations, known as Moran effects. And then big concepts in meta-community or in community ecology, like the meta-community paradigm, emphasize the role of dispersal and spatial dynamics in shaping communities. And this is an important process in the spatial synchrony of populations as well. But all that said, communities are more complex than populations, and maybe it's incorrect to assume that these effects on populations will percolate up to something that's felt at the community level. And certainly it would depend on us being able to measure the right things. So we've applied this approach to several different community level variables, but for now let's think about species richness. And we'll talk about how we go from data to statistical results here. So we start with the data array that contains time series of abundance or some index of abundance for multiple species across multiple locations. And for richness, we count the non-zero abundances to get to compress that array into a matrix of species richnesses for all of those locations through time. And then we calculate the pairwise synchrony between all of those locations and put that into a synchrony matrix, which has a lot of exciting information in its own right. And so we'll be kind of using that in the future to investigate some of these spatial patterns in synchrony of community properties. But for now, we're thinking about this big picture question of just, is there synchrony? And how does it vary across communities? So we're going to average that. And just to tease some of our results, as we apply this approach to spatial synchrony in species richness across several different grassland communities and several different coastal marine communities, looking at grasses and forbs in the grasslands, and looking at sessile organisms in those coastal marine environments. Among all of those communities, we see spatial synchrony and species richness ranging from near zero up to around 0.8, and the maximum on this this correlation is 0.1. So really some pretty high values here. Just to point out, our Jasper Ridge example that we keep coming back to is right in the middle here. So we see a lot of interesting variability across these communities, but one take home that I think is pretty convincing here is that species richness can exhibit spatial synchrony. And we are seeing some similar things when we look at other metrics like evenness. And so where to go from here? This is pertaining specifically to this subset of analysis, but I think looks a lot, or applies a lot, to the way that our group is functioning as a whole. We're going to analyze these gradients in community synchrony and empirical data to synthesize across many different communities in the LTER network, and then pair this with some theoretical work using simulations for insight into some of the underlying mechanisms, the ways of structuring communities and things of that nature that might give rise to different patterns of synchrony at the level of community properties.
And John, we had one question for you, which was why we're not using Kendall correlation, noted as potentially more appropriate for time series.
That's a good question. I don't honestly have a specific reason for not using Kendall correlations. I think the Spearman as just being a Pearson on ranks is a little bit more intuitive. But I would say that it would be applicable to also use Kendall as well.
And then we were also asked, can we have unequally spaced time series data?
If you're going to do correlations, correlation-based work, then having unequally spaced data is not problematic. As Lawrence elaborated with the wavelet statistics, then having some procedure for interpolating or filling in those missing values is really critical to being able to apply those tools.
And then finally, we were asked in reference to the wavelet analysis if the data were monthly, and I can start the answer on that. So for most of the sites, it was annual, and we tried to match it to the lifespan of the individual. So this led to a lot of discussion for aquatic systems that we're still working with. All of the results that we've shown for the grasslands were annually collected data. Lawrence, do you have more thoughts on that?
Yeah, I mean, just to emphasize that we were primarily interested in the kind of aggregate annual behavior of the community, particularly when we're looking at synchrony. We, apart from the fact that we might simply not have the relevant data to do it, just examining the monthly data it's completely dominated by the annual fluctuation in productivity that's associated with everything growing during the warm times and everything dying during the cold times. And that kind of annual fluctuation is a priori synchronized to the date. So it tends not to be very informative just to look for significant synchrony there. Whereas if we look at annualized data, we essentially filter out that annual fluctuation, and then we're just looking at variability in productivity or in populations from year to year, which has this kind of more interesting multi-year dynamics that we can examine. So yeah, so essentially we reduce the data to annual sampling by design.
And then we've had another question, which was that when some series are negatively correlated and another one is positively correlated, then that mean would be 0. So that would mean that the correlations might miss some patterns. And kind of briefly, we're exploring this, and John's been exploring a lot about thinking there can be geographic structure to synchrony. So you can have some areas that might exhibit high synchrony and others that might be very asynchronous or anti-synchronous. And that one of the things that we're finding, and I'll let John elaborate more, but it's not necessarily even proximal plots that exhibit positive, the kind of positive synchrony. So there can be a lot of geographic structure for a lot of different reasons. So we are doing explorations of that kind of geography of the synchrony as well as the mean. John, do you have more on that question?
No, I think that you really captured everything that I would have said on it.
Just one more point about communities. You know, often with spatial synchrony, we expect things to be positively correlated. We don't ordinarily expect to find negative correlation. I mean, there are circumstances, but it tends to be more with the community data that you might expect to see specific compensation between species. So we have we have many techniques, but we certainly have wavelet-based techniques for looking for specific negative phase relationships between pairs of species and testing for those. What we tended to find when we were testing individual species against one another was it was very difficult to find clear pairwise relationships between species. What we tended to find was we only really found interesting features when we were looking at the overall aggregate. So it's not necessarily the case that you have two species in competition and one of them is high when the other one is low. It seems to be more like the community as a whole compensates for the fluctuations that might occur in any particular member of that community.
All right. So it looks like we're having some more questions. I'm going to kind of quickly summarize like the review of what we've gone over, and then we'll open it up to the full questions free. But overall, what we've done is we've tried to apply a multi-pronged approach to understanding synchrony. And so one of the things we have found is really important is having frequency-specific variance ratio to highlight that synchrony versus compensation can occur at different time scales. And then using wavelet mean fields, we've been able to identify synchronous versus compensatory events over the time series. So seeing when in the time series those events happened and at what time scales they happened. And then finally, integrating these approaches have allowed us to understand synchrony and stability of communities as well as population. So that's an avenue that we've been continuing to delve into. And I also want to make sure that before before the hour's up, we'll be happily fielding questions, but just to make sure that we've acknowledged both NCs and the LTER. We are really grateful, especially Marty, Anna Peters, and Julian Brunn have done a lot to make this group be very smooth, and so we're very appreciative of that. And if you want to contact any of us, our emails are below. And with that, I'm going to go back to the other questions that we've been asked. So we've been asked, what is the appropriate way to calculate synchrony if I have the data, if the data were collected a few times in multiple years, so greater than 30 years, such as 10 or 15-day intervals in the growing season, for example, from May to September.
If I'm understanding that right, that's regularly sampled data. So you've got growing season data sort of every year for 30 years.
I think that's correct. So it sounds like they're doing, and please, questioner, correct me if I'm wrong, but it sounds like they're doing multiple, like high frequency intervals during the growing season for time series that were collected for many years.
So I just want to chime in and say, if people want to ask a question verbally, you can also raise your hand.
Oh,
do that
awesome.
Down at the bottom of the screen under More.
Lawrence, do you want to give a start to that question?
So yeah, if it's the case that you just want to aggregate that data over the growing season, then you've got you've got an annually sampled time series of data which is about the growing season, and it would be totally appropriate to use any of these correlation-based or wavelet or Fourier-based techniques to examine, the inter-year dynamics. So was this growing season similar to last year's growing season? Was this growing season, or was the average of this growing season, varying over a multi-year timescale? Is it varying over 10-year timescale, something like that? So these techniques would help you to tease apart that kind of inter-year dynamics. If you've got, I'm sort of rereading this question. If what you've actually got is a series of values from within a year, just over the course of a growing season, and then another series of values from the growing season of the next year, then you're probably not going to be able to, certainly using the wavelet and Fourier-based for timescale specific techniques, you're not necessarily going to be able to combine all of that data into one big wavelet or Fourier-based result. Because the problem is that obviously, although you have the data for the growing season, there's essentially what amounts to a whole load of missing samples for all the times that weren't in the growing season. And it really wouldn't make sense to interpolate those values or do anything like that. So in that case, I would say you would have to basically consider the growing season separately, possibly look at correlation-based analyses inside each growing season and see how they vary from year to year, and then aggregate the data to look at the inter-year dynamics. But there might be stuff you can do with correlation-based where you don't worry about timescales, where you could use all of that data, you know, you could bin all of that data together to look at correlation-based kind of approaches to synchrony. And you wouldn't have to worry about regular sampling then. So all of that data would be relevant to one big measure. Does that help at all?
Lawrence, could I just follow up with a question for you?
Sure.
Kind of related to what you were just speaking on, are there cases where it would make sense, you know, maybe you have like an annual, you know, completely annual grassland system. Would it make sense to set those abundance values outside the growing season to 0 and only look at the wavelet components for, you know, the sort of like the right time scales where you actually have within growing season measurements, and then later on, you know, those kind of across multiple years types of events?
Possibly. Essentially, what you're doing then is you're looking at a time series with a huge annual fluctuation in it. So like I was saying before, if you set all the non-growing season values to 0, your time series is just a massive annual fluctuation with details, basically. So when you do your, say you do your wavelet analysis or whatever, you're going to get an enormous feature at the annual time scale. And then whatever information is present in each growing season that you've painstakingly collected for those few weeks, will still be present in the wavelet transform, but it's liable to be completely washed out, I think, by that annual fluctuation. So I would still tend to think that it would make sense. I mean, essentially you're using the wavelet transform as a filter to look at different timescales, but it's not a perfect filter. And to me, it makes more sense to kind of pre-filter the data and look at the seasonal dynamics completely separately from the annual dynamics and the inter-year dynamics. But that's not to say that the information wouldn't be present if you did what you've just said, where you just set everything to 0 and include the annual fluctuation. It just might be harder to actually see what was going on and get something useful out of it.
So we have time for just 1 or 2 more questions from folks if they have them. Otherwise, I will say thank you to our panelists, and I'm sure that everyone out there in cyberspace is clapping for the to say thank you, but we can't hear it so well.
Well, thanks everyone for listening.
Yeah, thank you.